Morning, everybody. I apologize for the slight delay in starting the session this morning. The buses have taken a little longer to get through to the conference center this morning. But I think it's probably time to start, so I'd like to welcome you all to this morning's keynote session. It's a pleasure for me to have been invited to chair this session, so I thank Galton and his colleagues for that. And it's even greater pleasure to be able to introduce the speaker this morning, Professor Hao Wu, who's from Harvard Medical School, where she currently occupies the Springer Family Chair and holds a position in the Department of Biological Chemistry and Molecular Pharmacology. So Professor Wu's work is centered on molecular immunology and particularly phenomena related to the innate immune response. Her remarkable work over recent years has focused on the large macromolecular complexes which arise during intracellular signaling associated with the innate immune system, and how the interactions involved mediate the transmission of information from receptor to effector. Complexes such as signalosomes, inflammasomes, and even necrosomes, in which amyloid aggregates play a physiological role, are all found to play an important part in her research. As might be expected, she uses a wide range of tools to probe the biological questions she is interested in, including, inevitably, cryo-EM, as well as crystallography. During her career, she has worked with the likes of Michael Rossmann and Wayne Hendrickson, and has accumulated a large number of prestigious scholarships, honors, and awards. So her talk today is on structural elucidation of supramolecular complexes in immunity. Thank you very much for making the effort to come here, Wu, Professor Wu.
Thank you very much, Richard, for the very kind introduction. I would also like to, this is very high here. I have to use my tippy toes. And thank you very much for coming to this session today, especially given the competing sessions. So I really appreciate it. And please, make this very casual. You can actually stop me anytime. So indeed, my lab has been interested in using the tools of structural biology to probe fundamental questions in immunity. And this is reflected in our work over the years on TNF receptor signaling, toll-like receptor signaling, and inflammasomes. And in more recent years, we also started a program on adaptive immunity, in particular trying to address the molecular basis for the generating of antigen receptor diversity through the RAG recombinase. So in today's talk, I'm going to first focus on this macromolecular complex known as inflammasomes, which are caspase-1 activating machines in the cytosol. And you may know that there are many different caspases, but caspase-1 is a little bit different from those that involves in apoptosis. And the difference is that caspase-1 seems to have just very few defined substrates. And one is the pro-interleukin-1 beta, and the other is gasdermin D. And I will talk about how these molecules are involved in the inflammasome signaling. I will then talk about the effector in the downstream of inflammasome activation, that is how the gasdermin D molecule is involved in inducing pyroptotic cell death. And in the last few minutes of the talk, I plan to talk about the RAG recombinase in VDJ recombination. So why inflammasomes? One of the main reasons that the field has been chasing this complex known as inflammasomes is that the cytokine that inflammasomes activate, which is linked to interleukin-1 beta, is really the master regulator of inflammation. It can induce all the cardinal signs of inflammation that were initially identified more than 2,000 years ago, and that includes heat, redness, swelling, and pain, as exemplified here as a inflamed big toe in gout. So gout is actually a bona fide inflammasome disease because of high elevated level of uric acid and uric acid crystals induce inflammasome activation. So about 15 years ago, the first inflammasome was identified by the late York Chops group. And this really initiated the field. And all the canonical inflammasomes we now know contain a sensor component, an adapter component, and the effector, either caspase-1 or other related caspases, including 1, 11, 4, and 5. And the consequence of inflammasome activation is the conversion of cytokines, 1 beta and 18, and the induction of periapoptotic cell death. So there are now quite a big family of different inflammasomes, and a few that my lab has worked on, including the AIM2 inflammasome, which is activated by double-stranded DNA that is carried in either by invading bacteria or invading virus. Another form of inflammasome is the NLRP3 inflammasome, which is activated by a large array of stimuli, including uric acid crystals, as I mentioned, extracellular ATP, and bacterial toxins such as nitrorhizine. Somehow, these stimuli induce the activation of NLRP3. And another inflammasome that my lab has worked on is this NAPE and NLRC4 inflammasome. In this case, the direct sensor is very clear, and they are bacterial proteins that are part of the type 3 secretion system. So inflammasomes are becoming more and more important in medicine, not only in gout and other fever syndromes, but also in common diseases such as diabetes, cancer, inflammatory bowel diseases, and cardiovascular diseases. And it reflects a sensing of these inflammasome components to both microbial attack and sterile stimuli. So how about structurally? So in terms of the domain organizations of the AIM2 and NLRP3 inflammasomes, they are characterized by the presence of these domains called PYD, pyrine domain, and CAR domain. And both pyrine domain and CAR domain belong to the so-called DES domain superfamily, which structurally are very simple, but they form, as we showed earlier, they form these helical assemblies through concerted interactions between 3 types of different interactions. So when we first reconstituted the AIM2 inflammasome, we found that they form these star-shaped structures as shown here by negative stain electron microscopy. And if you label the different components in the AIM2 inflammasome, you see that the center is labeled, in the center are the sensor proteins, which is AIM2, or the adapter, which is ASC, whereas these long filaments actually all represent caspase-1. Also, if you look at the stoichiometry between these components, the AIM2 is always very little, with a little bit more ASC and a lot more of caspase-1. And if you think about this in the signaling scenario, this represents an amplification of signals from the sensor to the adapter and then to the caspase. So we solved the cryo-EM structures of quite a few of these filaments. I should note that most of these structures were solved using the Actica 200 keV machine, not the highest end machine that's available. So, so it turned out these filamental structures are related to each other through successive polymerization from the sensor to the adapter and then to caspase-1 through a PYD-PYD interaction as well as card-card interaction, as shown here through a fluorescence polarization assay. So, ASC on its own doesn't really polymerize very much, but if you add the AIM2 protein together with double-stranded DNA, which activate the AIM2 protein, you see a dramatic increase in the rate of polymerization of ASC. And similarly, you see the nucleation of Cas-based filament by the ASC protein. And this, at the time, really reminded us the situation with regard to cytoskeleton nucleation and polymerization, such as in the case for microtubule, the gamma-tubulin complex forms a 13-fold molecular entity that can then induce the polymerization of the microtubule. So our system really shares a lot of commonalities with other polymerization systems. So the model that we reached for both the AIM2 and the NLRP3 inflammasomes is this two steps of nucleation polymerization. As shown here for the NLRP3 inflammasome, the NLRP3 protein first form a platform, which then allows the PYD domains to nucleate ASC filament. And then the CAR domain on ASC further oligomerize Cas-based filament to promote dimerization of Cas-base and therefore activation of Cas-base. So even though this model is now well accepted by the field, as shown here by its adoption onto the cover of one of the most widely used immunology textbook, the question remains how these in vitro studies are directly linked to the cellular signaling complex that we see in cells. So here I show you an attempt to link the in vitro study to the cellular studies. So here is a reconstituted NLRP3 inflammasome visualized using electron tomography as in collaboration with Grant Jensen's lab. So, in cells, these inflammasomes remarkably form a single puncta, which we now realize, as shown here, that they reside exactly at the microtubule organizing center at the MTOC. And we're still, at this point, trying to understand why this localization and how microtubule dynamics can influence the activation of inflammasomes. So now let me switch gear to the third type of inflammasome that my lab has studied so far, and this is the NAPE and NLRC4 inflammasome. So in this particular case, for the NAPE2 inflammasome, the bacterial protein that NAPE2 recognized is this inner rod protein of the type 3 secretion system called PROC-J. It's a tiny little protein, and this is supposed to bind to the sensor protein that I'm showing you here, the NAPE2, which has this domain organization that contains a central ATPase domain followed by a leucine-rich repeat domain. So in this system, the adapter, instead of being ASC, is NLRC4. And this protein shares a fairly similar domain organization with NAPE2, again, starting from an ATPase domain followed by a leucine-rich repeat domain. So the question is that how do this complex associate with each other without the PYD-PYD interaction? Does the ATPase domain mediate the interaction between the sensor and the adapter? So we reconstituted the NAPE2 inflammasome in vitro by co-expressing three proteins, which is the ligand, the sensor, and then the NLRC4 adapter. We removed the N-terminal CAR domain in NLRC4 to avoid aggregation through helical polymerization. So what is quite surprising is that when we fractionated the complex, we see that the stoichiometry between NAPE2 and NLRC4 changes by the at the different positions of the sucrose gradient. And the best fraction here contains a lot more of NRC4 and a very little bit of NAPE2. And this is the only fraction that shows these complete ring-like structure as shown here by negative stain EM. So that gave us the hypothesis, perhaps, that in the complete ring of the inflammasome, most of the subunits belong to the adapter, whereas maybe only one subunit belongs to the sensor, because as you see here, very little of the sensor protein. And indeed, when we did gold labeling of the sensor, as shown here, most of the, or almost all of the inflammasome were just labeled by a single gold, suggesting there's only one copy of the sensor per inflammasome disk, again representing a signal amplification from the sensor and then to the adapter. So again, using the Actica microscope that we have at the Harvard University, in fact, material science facility, we collected data on these cryo-EM data on this complex and solved the structure using a 11-fold averaging. So in fact, these complexes are heterogeneous. We can see 10-bladed complex, 11-bladed, and 12-bladed complex. And the 11-bladed complex is the majority of it, and that's the one we were able to reach a decent resolution that we can fit the NLRC4 protein into each blade of the inflammasome. So this then gives us a way to look at the conformational change that occurs before inflammasome activation versus after inflammasome activation. So a crystal structure of the NLRC4 protein in the auto-inhibited conformation has been published a few years ago. And when we compare this structure with the active NLRC4 structure in our inflammasome disk, we see a dramatic conformational change at the LRR region, as well as in this associated helical domain here. Essentially, a 90-degree rotation occurred between the two forms that completely opened up the structure. As also shown here, this is the auto-inhibited conformation, and this is the active conformation. And the ATP binding site would be somewhere here. So there's a hinge motion between these two forms. So it turned out that this conformational change really unveiled a set of complementary surfaces that can then execute this polymerization reaction along the ring. So what do I mean by that? So if you look at the surface features between these subunits in the inflammasome disk, you see that they show a charge complementarity from positively charged and negatively charged. They form this nice interface in between. And this interface is completely absent in the auto-inhibited conformation. So here I labeled the two interfaces, 1A, 1B. So the A surface is completely buried in the auto-inhibited conformation, and it is the conformational change that exposes this interface. And therefore, this activation mechanism involves a ATPase-mediated conformational change that exposes the surface that allows the generation of the polymerization, as shown here using this movie. So the initial binding would kick open the structure, and which then allows the polymerization of the NRC4 adapter in a domino-like reaction. So this then leads to a model of NRC4 inflammasome activation through, again, successive steps of nucleated polymerization, starting from a ligand binding, which this step, some of these structural details we do not yet know, but this is more of a structure-based conceptual model. So the binding opens up the sensor protein, and then the sensor protein is able to successively activate more and more of the adapter protein. And at the end of it, the CAR domain that's at the N-terminal region of NRC4 forms a platform that can nucleate caspase-1 filament, and therefore reaching the end goal of the inflammasome activation, that is activation of caspase-1. So then these different types of inflammasomes that we have studied all utilize this nucleated polymerization mechanism. And the general concept is such that these multi-domain proteins are all auto-inhibited by intermolecular interactions before ligand interaction. And the ligand binding overcomes auto-inhibition, and at the same time provides, unveils or reveals a new binding site for the next molecule in the cascade to polymerize to generate a signaling. And you can imagine this process as a biological contraption that one step leads to the priming of the second step, and therefore a signal transduction as well as a signal amplification process. So these work on inflammasomes are in sync with some of the other work that we did previously, which showed that these aggregated forms are really the functional forms, signalosomes or higher order assemblies, and these include these helical assemblies that we have seen in multiple systems, multivalent assemblies that can be generated as long as you have multiple oligomerization domains in the same protein. Then you can imagine a cross-link reaction to generate a higher order structure. Also, we have observed these amyloid assembly that is not mediating disease aggregate, but rather generates a platform for kinase activation to induce a signaling process. So these studies over the years, I think, has really provided a connection between the cellular signaling clusters that are in the micrometer scale with the underlying molecular structures that are in the nanometer scale. And by bringing all the effector enzymes, such as caspase or kinase, into proximity, you increase the local concentration dramatically, and this then promotes the weak allosteric interaction that can happen in this local high concentration to drive the activation of these enzymes. And this also explains that many of the signaling processes in innate immunity has a feature of threshold response that is either no response or a full response in a digital fashion. So a few years ago, we proposed that these higher order assemblies that we observed and also other people observed in innate immunity represent a new mode of signal transduction. In fact, this slide is a little bit old, a few years ago. So at that time, EGF receptor was classified into the classical mode of signal transduction that is elicited through a conformational change followed by activation of kinases and other effectors to generate a cellular response. But more recently, even EGF receptors, RAS signaling, are now also appears to form higher order structures. So we expect that, or we predict that really more and more biological systems may utilize these large molecular machines to execute their signaling functions. So how about then the downstream effect of inflammasomes? So as I mentioned a little bit in the introduction, there are two key events that are induced by inflammasomes. First is the processing of cytokines and its release to the outside of the cell. And the second is a cell death known as pyroptosis. And this is a form of cell death that essentially breaks up the cellular membrane so that cellular contents can be released to generate an inflammatory kind of cell death. And this is different from apoptotic cell death, which is usually more contained and less inflammatory. So these two events, it turned out, are highly related. And this came from work both from our lab and many other labs, including Feng Shao's lab and Vishwar Dixit's lab. So in this schematic, caspase-1 or caspase-11 activation leads to processing of the cytokine. But these cytokines are leaderless peptides. They cannot be secreted through the classical secretory pathway. So how do they get released to the outside of the cell? So this really depends on the processing of this protein, gasdermin D. And this protein, upon cleavage by caspase-11 or caspase-1, its N-terminal fragment somehow cause this lytic cell death. And this lytic cell death then allows the exit of the cytokine at the same time. So we investigated how this N-terminal fragment of gasdermin D can induce cell death, a lytic form of cell death. So it turned out this N-terminal region can oligomerize on cell membrane, as shown here. And the reason that it does this is through a specific affinity to acidic lipid, as shown here by these lipid strip binding assay. So gasdermin D, and only the N-terminal fragment of gasdermin D, but not the full-length gasdermin D, can bind to PIPs, which are acidic lipid, PIPs, phosphatidic acid, phosphatidylserine. And you may know that all these acidic lipids reside to the inner leaflet of the plasma membrane, only the inner leaflet. And this means then gasdermin D can only kill the cell from within, from inside the cell, but not from outside the cell. And gasdermin D can also interact with cardiolipin, which is a lipid in the mitochondria and in bacteria, which suggests that gasdermin D can also kill the invading bacteria inside the cell. So indeed, if we reconstitute a liposome with these acidic lipids, then when you add gasdermin D together with a caspase, you see a leakage of contents from the liposome, which we have shown with this liposome leakage assay. And if you reconstitute gasdermin D into the liposome, you see these pore structures. And you can solubilize these pores, and they all look like this. And if you look at the inner diameter, which is somewhere between 15 to 20 nanometer, they are definitely large enough for the passage of processed interleukin-1 beta, which is probably around 5 nanometer in diameter. So then explaining that these pores can act as a portal of secretion for the cytokine. So just very recently, we obtained the cryo EM structure of the gasdermin pore, as shown here. You can see at this resolution, roughly about 3.8, as reported by Relion, you see these individual beta strand that forms this gigantic pore structure, a total of 108 gigantic beta barrel. So we're still analyzing the structure, but what you can see is that already the gasdermin structure form these beta strands in this pore form, whereas in the crystal structure of the inactive gasdermin. This is the C-terminal part, which get released after cleavage. So the N-terminal part is here. It has much fewer beta strand contents, and there's a huge conformational change between the inactive form and the pore-forming form. In a way, we're still analyzing the structure, but in a way, this is, looks like it's quite similar to these bacterial toxins, pore-forming bacterial toxins. So the last 10 minutes or so, let me switch gear to the RAG recombinase. And this is the enzyme that executes the first crucial step in VDJ recombination. So you may know that in the germline lymphocyte, you actually do not have an immunoglobulin gene. All you have are these individual segments of VD and J, and they need to be spliced recombinatorily into the real Ig gene during lymphocyte development. And this is a process that's incredibly powerful, and it can generate a diversity that is much higher than, I think, any of the known display libraries to date. So the RAG protein, which is a hetero complex of RAG1 and RAG2, is associated with some of the most severe form of immunodeficiency, as shown by the so-called bubble boy syndrome. And it is also related to cancer and other forms of autoimmunity. So what RAG does is to generate a cleavage in the VD and J gene. And then the second step is taken over by the DNA repair machinery so that they can be spliced together. So RAG does this by recognizing the so-called recombination signal sequences, RSS. And even though RAG is considered considered similar to recombinases, it is different from most recombinases by recognizing two different sequences. One is a 12 RSS, the other is 23 RSS. So how is this defined? So in both RSS, there is a conserved heptamer sequence. This is a sequence. This is a sequence that is specifically recognized by RAG. Heptamer, and then in 12 RSS, you have a 12 base pair spacer in between, followed by a nonomer sequence. So basically, you have two sequence-specific, you have two sequences that are specifically recognized by RAG, followed by a region that is not specifically recognized, either a 12 base pair or a 23 base pair. And this is important because 12 RSS and 23 RSS flank different segments, VD and J. And because RAG can only recombine a 12 with a 23, the so-called 12-23 rule, by doing so, you only splice different kind of segments together, but not the same kind of segments. So the 12-23 rule is really a critical concept in ensuring the fidelity of VDJ recombination. Okay, so RAG generates this cleavage through two steps catalytically. In the first step, it nicks the double-stranded DNA at the exact junction between the coding region, which is either VD and J, and the signal region, RSS. And then this newly generated 3' hydroxyl would attack the opposing strand to generate a hairpin structure in the coding segment and a blunt-ended cleaved RSS. This part essentially it has no further function and will be circularized and linked and discarded But this part, the hairpin DNA, gets shunted into the repair machinery so that they can all get spliced together. So the RAG recombinase have the domain organization that I'm showing you here. RAG1 have all these different domains, but don't worry about it. Essentially, you have a DNA binding domain in here, and that's the core of the RAG1 protein. And for RAG2, it has a N-terminal WD40 domain followed by a C-terminal PhD domain. And the PhD domain is only important for its function inside the cell because it helps to recruit RAG to to active chromatin structures through its recognition of the H3K4 trimethylated sites. So RAG function not only require RAG1 and RAG2, but also require this protein HMGB1. Some of you may know that HMGB1 has two HMG boxes, box 1, box 2. And what HMGB1 does is to bind to the minor groove of a double-stranded DNA so that it generates a kink. And this is actually, the type of kink is actually quite similar to the structure that Steve solved many years ago. Is it TF2B? Yeah, okay, it's almost escaping me, yeah. Right, so a crystal structure of the RAG1, RAG2 complex was reported in 2015 from Wei Yang and Martin Geiler's group. Our goal was to look at the DNA bound forms and try to explain the catalysis as well as the 1223 rule. So we designed a number of DNA sequences. I think the most comprehensive one is this NICT RSS, which contains both the coding end. It starts with the coding end, and this is the nicking site, followed by the entire RSS that contains the heptamer, the spacer, and the nonomer. So I think we were pretty fortunate in this design because the entire DNA sequence were visible in the cryo-EM structure that I will show you shortly. So this cryo-EM data was collected using a polar microscope using a K2 detector. So the structure essentially contains a RAG dimer. It's the cyan here, and the light yellow or greenish yellow is RAG1. And RAG2 is this WD40 structure, this greenish thing and this bluish thing here. So what you can see already is that even though this is a dimer structure, the top part of the dimer does not align with the bottom part of the dimer, which is the nonomer binding site, the NBD, nonomer binding site, NBD. So the DNA, this is the 23 RSS in orange and 12 RSS in red. The DNA is highly kinked, and there's also other distortions that I will show you shortly. Highly kinked, and in fact, they are bound to HMG molecules, as I will show you. And this is the catalytic center here, and this is at the highest resolution of 3.3 angstrom. We can clearly see the metal ions and all the catalytic residues lined up to perform the catalysis. So here, you can see some of the bases that's shown in dark color that are actually flipped out of the double-stranded DNA. And that's part of the DNA distortion that are important for RAG, because RAG has to catalyze 2 steps of catalysis with the same active site. And the distortion allows the position of the nucleic acid to the vicinity of the active site in both steps of the reaction. So the DNA distortions include base flipping at the heptamer site, as shown here, also at the coding end. You see the cryo-EM density is really quite clear in the DNA region. You can see exactly how the bases are flipped out. So if we compare the DNA-bound form of RAG versus the apo form of RAG, you see a fairly dramatic conformational or fairly dramatic closure of the dimer upon DNA binding. As shown here, you see this up to 27 angstrom movement upon the binding, and also shown here in this morph between the apo structure to the bound structure. There's a closure. And each DNA is bound. This is actually, I think it's important. Each DNA is bound by both monomers of the RAG dimer. So that indicates there will be a cooperative conformational change that's induced by DNA binding. So, how were we able to visualize the HMG boxes? So we first put in all the protein residues as well as DNA that we can see. And then what's left are these fairly low resolution blobs next to the DNA. So fortunately, even though I think that region is probably barely 7 angstrom or maybe even below, a number of helices or just tubes of density are visible. And because the HMGB box structures are known, we were able to fit these hairpins of helices into the density and therefore explain these things as HMG boxes. And in this case, the 12 RSS, which is the one with the shorter spacer, we can observe one HMG box, and that induced a 60-degree bend in the DNA structure, whereas for the 23 RSS, we see two HMG box binding to the side of the 23 base pair spacer, and that gives a 120-degree bend. So this, I mean, I've never seen anything as dramatic as this, in terms of the distortion in the DNA. So then this led to our structure-based model, that is, this induced asymmetry as well as the simultaneous bending of the bound DNA is the molecular basis for the 12-23 rule. So as soon as one side of the RAG dimer binds to a 12 or a 23 RSS, the MBD region tilt to one side or away to the other side, and therefore the other side can only accommodate the other types of RSS, but not the same kind of RSS. So we currently will continue to work on this project, and in particular, we're interested in populating additional structures in this proposed catalytic pathway, and we're doing this in collaboration with the New York Structural Biology Center using Bridget and Clint's Titan Creoles. So in summary, I talked about our work on inflammasomes, including these DES domain mediated filamentous structures, these ATPase domain mediated disc-like structures. So I should add, perhaps, it is known that these ATPase domains can form all kinds of ring-like oligomers. But I think in our case, this polymerization that's mediated by the ATPase domain has not been observed previously. That is, once you activate one, you can activate successively the rest of the subunits. And this nucleated polymerization mechanism seems to become a overarching principle in inflammasome assembly and also in many aspects of innate immunity. So in the case of gasdermin D, there's actually a big family of these proteins now, and they appear to be involved in the induction of different kinds of lytic cell death in response to different kind of stimuli. And we think that this may become a very important aspect of cell death, cell death in response to diverse insults. Okay, so the work was performed both by my talented postdocs as well as by a number of wonderful collaborators. So the inflammasome projects, quite a few people in my lab spearheaded the different aspects of the project. Alvin Liu was a former student. Jianbing, Vanny, Liman, they're all still in my lab, but some of them are now on the job market trying to establish their own labs. Heng spearheaded the RAC project. The inflammasome project, the filamentous structure part of the work was initially in collaboration with Ed Egelman. We are now also doing a lot of it ourselves. The NRC4 inflammasome was done extensively together with Jack Mao's group, and the RAC project was a close collaboration with Mao Fu Liao's group at the medical school. And the Gasdermine D project is a collaboration with Judy Lieberman's group in our department. And I thank you for your attention.
Oh,
okay, thanks.
Could I just, first of all, just thank her for a fantastic talk, and go ahead. Oh, this one doesn't work, sorry.
I didn't hear what you said, sorry, but I presume I can ask a question. So beautiful stuff, Hao Wu.
Thank you very much.
The Gasdermine D structure, the pore, were they distinct sizes, those pores, or did you see variability?
Yeah, there are variabilities, but thanks to Cryo-EM, we're able to distinguish them. The different, yeah.
So how much variability was there?
It's not huge, actually. Oh, so I should say that's not Gasdermine-D, so that's A3 as well. Okay. It's A3, yeah. So the variability that we can see from 2D averages between 26, 27, and 28. Okay. So luckily, I think that's a fairly small range, and the 27 is a major population.
And I don't think you specifically said, but the beta sheets that extend, do you think they're penetrating the membrane?
Oh, yeah.
And
yeah.
They're the right size to...
They're about the right size, yeah, right. Mm-hmm. We can also reconstitute them in nanodisk. Yeah.
So perhaps just carrying on from that. So is there any selectivity with respect to this pore? I mean, the size of, you talked about...
It's huge, yeah, right.
So it's basically, everything's going to leak out.
A lot of anything that's,
So there's nothing really special about interleukin-1 beta in that respect, or is there?
Yeah, that's what I think. Like LDH, a lot of people use that as an indicator of cell death. That obviously is also small enough to pass through the pore. We don't really know, at least in vitro, I think we have done this together with John Kagan in our department. So we try to use different sites of polymer, whatever, yeah, to see the selectivity. So it's, right now, at least it looks like it's just a selection of size. But it could be much more, it could be more complex in vivo. Yeah.
Okay. Any further questions? Yes?
Thank you for an amazing talk. Would you like to give some comments about how to purify or express these amazing complexes that will give us beautiful super complexes?
Yeah, well, it's probably better to ask my postdocs. But it is, I would say that's the major challenge, to reconstitute them. I mean, in my lab, we do various different expression systems, co-expressing them in insect cells, in mammalian cells. We seldom use bacteria nowadays. It's almost everything are insect cells and mammalian cells. It gets extremely expensive. Yeah, I mean, but I think nowadays, even much more complex, system that you have even more number of components can be reconstituted. Like yesterday there was a session on protein expression. I think they all achievable at this point. I think you just have to try it. Yeah.
And any other questions? Just going back to the first part of your talk.
Okay.
Maybe this is one of the aspects which you said weren't fully elucidated, but what can you say about the way in which the conformational changes triggered in the sensor protein as a response to ligand binding. Is much known about that?
You mean the NRC4 inflammasome?
Yeah.
Yeah, okay. Yeah. So if you look at the auto-inhibited conformation, well, in the active conformation, I said there's a A interface and a B interface. So the A interface is completely buried. But the B interface is similar, but not exactly the same as in the active form. So what I imagine, I think this would require single molecule studies. What I imagine is that this partially formed B interface get kicked by the activated sensor protein. And that, reset this interface to be the form that's in the open form, and at the same time opens up the thing.
Okay. But
you see what I mean? Yeah.
It wasn't entirely clear to me if you know where the ligand
ligand,
binds.
no,
Yeah.
we don't know that. So that we're still trying to get the structure with the ligand. Right. So we still don't know that. No, that's a missing part as well. Yeah, right.
Okay, thank you. Thank you very much. Any further questions? If not, then perhaps we could thank Hao once again for the great talk. And I think just before everyone disappears, someone from the local organizing committee would like to say something.
Oh.
Can you please come on the stage? Hi.
Oh, hi.
So it's my great honor to present a small token of appreciation on behalf of LOC.
Thank you.
Thank you
yeah.
very much.
Thank you. Yeah. Very nice. Yeah. Yeah. Thank you. Thank you very much. Yeah.
Thank you.
Even a present. Oh my goodness. Yeah.
Even I got a present.
Even you got a present. Yeah. Maybe yours is better.
Thanks for everything. That was great.
Oh, thank you. Yeah.